I'm Dr. Leslie Blankenship-Williams, and we are going to finish our introduction to endocrinology by learning about the chemical classes of hormones. Now, the chemical classes of hormones is an important topic because understanding the chemistry of the hormones allows us to predict certain aspects of their behavior in our body. So let's first introduce the three classes of hormones from a chemical standpoint. They are steroid hormones, which are derived from cholesterol, peptide hormones, which are going to be short chains of amino acids, ranging anywhere from 7 amino acids to 300 amino acids, and then amine hormones, which is actually a single amino acid that has been modified. So go ahead and write these down. These are three classes of hormones. So that means that some hormones are actually built from cholesterol, which is a steroid or a type of lipid. Some hormones are chains of amino acids linked together and then folded into some structure. And then some hormones are really small. They're just a single amino acid that's been structurally modified. So we're going to start by looking at the steroid hormones. So steroid hormones are all derived from cholesterol. We have a tendency to think ill of cholesterol and think it's bad, all cholesterol is bad, because of its effects on our low-density lipoprotein and the outcome of that, which is the buildup of atherosclerosis or arterial plaque. But cholesterol serves a super important function. Not only is it vital to our plasma membrane structure, it's also the starting material for all steroid hormones, and there are plenty of steroid hormones. So you can see here we've actually got three different steroid hormones listed: aldosterone, cortisol, and estradiol, which is a classic estrogen. I'd like you to take a quick look at these and particularly look at aldosterone versus cortisol and see if you can spot the differences. You'll notice that the 4 fused ring structure is largely retained from molecule to molecule, but what is hanging off of those 4 rings differs. And aldosterone and cholesterol have very few differences. It feels like one of those games. Spot the differences between these pictures. And so you can see here the difference is right there versus right there and right here. Very, very small. So when we talk about modifying cholesterol, they may not be huge modifications in order to create a new hormone. Now, all steroid hormones are derived from cholesterol. If you are in my class, you absolutely do not need to write this down, but I wanted to just point this out to show you the pathway that we always start with cholesterol and it gets modified into another hormone that then can go different pathways. Notice if we go down, we can create progesterone, we can create corticosterone, we can create aldosterone. But if we take progesterone and instead go this way, we create cortisol, or we can create testosterone, another type of testosterone, and then estrogen. So you can see that by continually modifying cholesterol and its derivatives, you can create a variety of steroid hormones. Steroid hormones are really only secreted from 2 places in the body. They are the adrenal cortex. So if you've been through anatomy and you've seen the cadaver, the adrenal glands are poorly defined often because they feel like they're adipose tissue, they feel fatty, and that's because the adrenal cortex is secreting large amounts of steroids, which are lipids. So it feels like a lipid-rich organ, and it really is. And then the other ones are going to be the gonads. Those are going to be the testes and the ovaries. Okay, let's take a look at the biggest class of hormones, and that's going to be the peptide hormones. So peptide hormones are going to be a chain of amino acids. It can be a short chain, like 7 amino acids, or it can be a pretty long chain, like 300 amino acids. On display, I have some peptide hormones that are involved in the reproductive cycle. Oxytocin you might recognize as the hormone that induces labor and delivery. And if you happen to work in the labor and delivery wards, a common synthetic version of oxytocin is going to be Pitocin. So if you want to induce labor and delivery, you might start giving a very pregnant woman Pitocin through an IV drip. Luteinizing hormone, follicle-stimulating hormone, and human chorionic gonadotropin are all going to show up in a subsequent lecture. But what I wanted to show you here is how similar they are. Luteinizing hormone, in fact, and human chorionic gonadotropin look very, very similar, which I think is interesting. Just to go off on a little bit of a tangent, luteinizing hormone is going to be used to induce ovulation. And in fertility treatment, so if you're trying to work with somebody who is going to produce even more eggs than they typically would in a cycle through hormone manipulation, they actually use human chorionic gonadotropin as a substitute for luteinizing hormone because it looks so similar. Because it looks so similar, it can actually bind to the same receptor. I get ahead of myself here. This is coming in reproductive endocrinology. But all I wanted to show you here is that we have a variety of peptide hormones. Again, we're linking together amino acids and creating a specific folded shape. Our final chemical class is going to be the amine hormones. Now, all amine hormones are actually derived from the same amino acid, and that is tyrosine. In my class, you do not need to memorize that fact. But if you're studying for the MCAT, that might be useful. So tyrosine is the amino acid that is then modified to create a variety of hormones. You should probably recognize some of these. We have dopamine, epinephrine, and norepinephrine. And notice how they are all very similar to tyrosine, but even more, they're all similar to each other. And then on the right side, we have thyroid hormones, and there's two of them. You might recognize that the thyroid hormones, which go by T3 and T4 respectively, have iodine attached to them. That's kind of unique, and one of the sole places that iodine is needed in our body. And in fact, we tend to fortify our salts with iodine to make sure that we have enough to create our thyroid hormones. So definitely write down that the amine hormones are going to be broken down into two subcategories. The group that includes dopamine, epinephrine, and norepinephrine are all called the catecholamines. So write that down, catecholamines. Epinephrine, its partner in crime, norepinephrine, and then dopamine. And in a previous lecture, we looked at the similarity between dopamine and norepinephrine, and therefore how an antidepressant that should only affect dopamine secondarily affects norepinephrine because of how similar they look structurally. And if you look here, again, it's the spot the difference. What is the difference? Well, the only difference is right there. The thyroid hormones, you do not need to write out their full name, just recognize T3 and T4. What's more important is that you recognize the catecholamines and thyroid hormones are both subcategories of the amine hormone. Now, what we are going to do at this point is create a giant table that looks at the properties of steroid, amine, and peptide hormones. So I'd like you to first pause the video and copy all of this down. Okay, now let's go ahead and start discussing this. We'll go ahead and start with what we already know. We know that steroid hormones are made from cholesterol. We know that amine hormones are modified tyrosine amino acids, and we know that peptide hormones are going to be short chains of amino acids. So go ahead and copy that down. And now we're going to take a look at the new stuff. The way I'm going to approach this is I'm going to talk about steroid hormones and I'm going to talk about peptide hormones. And I'm going to leave the middle column, the amine column, blank for now. So really we're just looking at two categories, steroid and peptide, because they tend to be opposites, because they behave differently in the body. So let's go ahead and start with what a steroid hormone is. It's cholesterol, or a derivative of cholesterol. Cholesterol is a lipid, and lipids do not like water. So because they do not like water, they're hydrophobic. When you throw a steroid hormone out into the blood, it doesn't want to be there. It's like, oh, water, water everywhere. Water is scary, right? So it is poorly soluble in plasma. Peptide hormones are made of amino acids, and while the residues of amino acids can be nonpolar, generally the backbone is very polar, likes water, hydrophilic. And so peptide hormones tend to be hydrophilic, and they tend to freely dissolve in plasma. So let's go ahead and write that down. Now, the fact that steroid hormones are not soluble in plasma and peptide hormones are has some consequences. If steroid hormones are not soluble in plasma and you throw a steroid hormone out into the plasma, what's it going to do? It's going to start freaking out and need to find the nearest safe space. Well, what is the nearest safe space? Most certainly a plasma protein. So in a previous lecture, we learned that a lot of lipids are transported, protected by these plasma proteins that kind of like wrap themselves around and hold it tight and protect it as they run around the plasma. And they kind of shield that lipid from the scary water. And the same is true for steroid hormones. Steroid hormones are transported by plasma proteins in the plasma so that they don't have to interact with water, whereas peptide hormones are not. They don't need to be. They are freely dissolved in water, and they're like, I don't need any help. I'm fine out here. So go ahead and write that information down. Now, the fact that steroid hormones are going to be carried and protected in plasma proteins where peptide hormones are free also has a consequence. There's something called half-life, which is a very important measure of how long that hormone lasts in the body. It's not indefinite. Every time you secrete a hormone out, there are enzymes out there trying to chop that hormone up and break it apart so that the hormone becomes deactivated. This is needed. Can you imagine right now if you like almost got into a car accident, maybe did get into a car accident, and your epinephrine is pumping through your body because you're in fight or flight mode, and it stayed there indefinitely? And so, I don't know, 8 days later, the fear and threat of the car accident has long since passed, but you're still freaking out and having a fight or flight reaction because the epinephrine is still circulating in your body. That would be bad, really bad. In fact, you only want that response to be there as long as the danger is there. So you want that hormone to be broken down really quickly. So peptide hormones, because they are freely dissolved in the plasma, are accessible to those enzymes that are just waiting for that hormone to come by and break it apart. Therefore, peptide hormones are rapidly broken down. You secrete a peptide hormone into the blood and it gets rapidly broken down. Steroid hormones have a longer half-life. That means when you secrete it into the blood, they end up getting protected by plasma proteins, so it's harder for enzymes to find them and break them down. And so because they are hidden, enzymes take longer to break them down because they're harder to access. And therefore, steroid hormones tend to have a longer half-life. Now, by half-life, I mean this. If I secrete 100 hormones into the blood, how long until I only have 50 left? That's the half-life. So for a steroid hormone, it's usually like 30 minutes or more, anywhere between 30 minutes to a couple hours. For a peptide hormone, it's usually under 10 minutes. There's some exceptions, but it tends to be shorter. For our purposes, we're just going with the generalities here. So we're going to say the half-life for steroid hormones is high, meaning it's long. And so 30 minutes to hours. For peptide hormones, it's short. Peptide hormones are rapidly degraded, and therefore they don't last very long in the blood. That's actually a good thing. That means that if the peptide hormone, once you secrete it and it gets rapidly degraded, you can assess whether or not you want to secrete it again. Like, oh, do I still need this hormone? If so, I'll secrete more. If not, problem solved, and I don't have to deal with, an abundance of that hormone when I no longer need it. So hopefully I've conveyed that having actually a short half-life is good for control of that hormone level. So now we're going to take a look at the receptors. Now I've already talked about the fact that a target cell must have a receptor. And when I drew that target cell in the previous lecture, I put the receptor facing the extracellular fluid. So you had this green hormone and it was going to bind to this receptor. Well, not all receptors are actually facing the extracellular fluid. In fact, some receptors are located inside the cell. So we do have a number of receptors that are called extracellular receptors, the word extra outside, and those are going to be the ones that are embedded in the plasma membrane, and their face, where they bind, faces the extracellular fluid. But we also have receptors that can be located inside the cell. And if there is a receptor inside the cell, whether it's in the cytoplasm or the nucleus, we call that an intracellular receptor, intra within. So what does this mean? Well, steroid hormones can use both intracellular receptors and extracellular receptors, where peptide hormones tend to only use extracellular receptors. So let's go ahead and add that into our chart. So the rationale for why steroid hormones can use intracellular receptors where peptide hormones don't is not something I'm going to cover. So our next question is, are these hormones synthesized on demand or in advance? And let me give you an example of that. Let's say that I run an Etsy shop and I make face masks, actually. It's actually true, I do make face masks. So let's say I make these face masks. If I were to make all of these in advance, I would make like 10 of these, and then once I had 10 of them, I would list them on Etsy. An alternate business model would be to synthesize on demand. I would make sure I had the fabric for 10 face masks, but I would just list 10 on Etsy, but I didn't make them. When somebody buys one, then I will stitch one together and mail it off. Which one is faster? Well, in terms of the person getting the face mask, obviously me making it in advance would ensure that I could rapidly package it up and send it off. If I have to synthesize it on demand, there might be some lag time. So of the two, peptide hormones have the advantage in that you can make them in advance, store them in a vesicle, and then when that cell, the endocrine cell, is told to secrete, you just move that vesicle up to the plasma membrane, fuse it, and you have exocytosis. That secretion, you just secrete it out. It's a very rapid response. Steroid hormones don't have that luxury. And the reason is because steroids are lipids. And if you try to synthesize a bunch of lipids and corral them into a vesicle, you're bounding them by a plasma membrane. Well, guess what likes the inside of that plasma membrane? Steroids. And so the steroids have a tendency to leak out. In fact, they can kind of leak out of the cell. And so you can get leaching of these steroid hormones that you've made out of the cell if you make too many of them. That's bad because remember, steroid hormones are effective in really small concentrations. So even leaking of steroid hormones out of these cells would potentially have disastrous consequences for us. So as a byproduct, the way that we work this is we simply just make them when we need to. When we need to, we take the precursor and we just modify it into whatever it is we need. So steroid hormones are going to be made on demand, and peptide hormones are made in advance because you can corral the peptide hormones, which are hydrophilic, in a vesicle and keep them. My last category is genomic effects ? So in other words, what types of responses do these hormones tend to exert? This one isn't nearly as clean as the others, so I tend to talk about this in very general terms. Genomic effects means to alter the DNA, so you're expressing new genes and therefore you're making new proteins. Steroid hormones largely have genomic effects. They don't all have genomic effects, but it tends to be a trend. Some peptide hormones do as well, but it's not as common. So we can summarize this by saying that steroid hormones often exert genomic effects and peptide hormones sometimes exert genomic effects. Okay, so now that we've covered all of this, let's come back to our middle column, which is the amines. So what are we going to do with those? Well, the reason I saved it to the end is because I'm going to give you a shortcut. I've already covered quite a bit, steroid hormones and peptide hormones. Some of it you have to memorize, but some of it, if you remember that cholesterol is a lipid and doesn't like water, and peptide hormones do like water, you can pretty much figure out what's going on. Now, what we can do with the amines is we can go ahead and draw a line. Okay, so for the amine hormones, we can actually cheat a little bit and create a shortcut to help us remember how the amine hormones behave. We're going to draw a line because remember there are two subcategories of amine hormones. We have the thyroid hormones and the catecholamines. Well, it turns out that in most cases the thyroid hormones behave just like steroid hormones, whereas the catecholamines behave just like peptide hormones. So rather than having to memorize 4 columns, we just need to memorize 2, what the steroid hormones are doing and what the peptide hormones are doing. Then copy and paste all of your knowledge for steroid hormones over to the thyroid hormones. And copy and paste all of your knowledge of the peptide hormones over to the catecholamines. Now I'm going to show you a picture from your textbook, and I've kind of modified it a little bit. It has on the far left the peptide hormones, in the middle the steroid hormones, and then I've drawn arrows to show the catecholamines mimic the peptide hormones, and the thyroid hormones largely mimic the steroid hormones. So all you need to know for my class is the peptide hormones and the steroid hormones, and then copy and paste the catecholamines with peptide and thyroid with steroid. Okay, our final question is this. If I give you a hormone, let's say aldosterone, many of my test questions ask you, is it a steroid hormone, an amine hormone, or a peptide hormone? And you might be thinking to yourself, oh goodness, I don't want to have to memorize all of this. But I'm going to tell you some tricks, because there are some tricks to figuring out which ones are which. Here they are. Get ready, write this down. Steroid hormones almost always end in one, O-N-E, or ol, O-L. In other words, the name of the hormone ends in on or ol. So progesterone, estradiol, testosterone, aldosterone, cortisol. So largely, I mean, it's not 100%, but largely, if you see on or ol, it's a steroid hormone. For my class, all the hormones that I might give you that are steroid hormones are going to have on or ol at the end of them. The amine hormones, you just have to memorize. Know the catecholamines, dopamine, epinephrine, and norepinephrine. And the thyroid hormones, T3, T4. Everything else is a peptide hormone. Everything else. So let me give you an example. What is vasopressin? And you go, I don't know. We haven't learned vasopressin. Apply the techniques. Does it end in on or ol? No. It ends in in, so in, vasopressin. So it's not a steroid hormone. It's also not dopamine, norepinephrine or epinephrine, or a thyroid hormone, so it's not an amine hormone. So therefore, it must be a peptide hormone. All right, what's aldosterone? Aldosterone ends in O-N-E. It's a steroid hormone. What is epinephrine? Epinephrine is one of the amine hormones that you just have to memorize. What is erythropoietin? Well, it doesn't end in on or ol, so it's not a steroid hormone. It's not dopamine, norepinephrine, epinephrine, or the thyroid hormone, so it's not an amine hormone, so therefore it must be a peptide hormone. So whether you're in my class or you're studying for an exam, hopefully that trick will help you remember which chemical class a hormone belongs to. And with that, we have concluded our survey of Introduction to Endocrinology. In our next lecture, we're going to explore how glucose is regulated in the blood. I'll see you then.